All right, everybody, welcome back to the Biochemical Genetics Clinical Course Seminar Series. This is week 7. It's August 30th, 2024, and we're welcoming back Dr. Nick AhMew from Children's National. Really happy to have him back and talk about urea cycle disorders and MSUD. Just some housekeeping. The code will be in the chat. It's BOMSAR, and I'll be copying it over for anybody who gets on a little bit later. Next week, we have Dr. Al coming back to talk about carbohydrate metabolism. As per usual, this is going to be posted in its entirety and archived on the SIMD website. So if you want to reference it later, it's free and it's downloadable. And you can put questions in the chat or camera on and full mic, anything you want. But well, Nick, I don't know what your preference is for questions during versus after, or we can
Oh
see.
no, I'm happy to take them as we go along here. Just the way I've organized my screen here, I may not necessarily see them. So, Pete, if there's like a backlog of questions or things like that, can you just let me know?
Yep, absolutely. And I'll give it over to you.
Perfect. Well, thank you so much, Pete. Again, it's always a pleasure and an honor to be able to give this talk. I would add that it's increasingly challenging every year because we learn a little bit more. And so this talk always, I try to add a little bit more, but we're given the same amount of time. So I'll try my best to cover here everything that I would like to in this time. And again, if there are questions that come up as we go along, please feel free to ask. Typically, my talk has been usually is the first talk in this series, but this year, due to my own scheduling, had to be a little bit further down. Just to share a couple of disclosures here in terms of grant support. For instance, I am a member of the Urocycle Disorders Consortium that is funded by the U54. I'm a consultant to several different ongoing clinical trials in different capacities in terms of data review. And then I'm a medical advisor to the NUCDF, the Cintron Foundation, the MSUD Family Support Group. So again, maybe this was already covered. Apologies if it already had been as part of the amino acid 1 talk. Mine is usually that talk. But I just want to briefly review, as you know, that protein is made of amino acids, and this is a basic amino acid structure here, that there are 20 different protein amino acids that are all necessary in order to be able to synthesize any type of protein. And that the fate of these amino acids is such that this nitrogen group has to be eliminated in some fashion when you're talking about breaking down amino acids. And this is actually accomplished differently depending on the type of animal species. So really interestingly, in most aquatic animals and in fish, that this nitrogen can be released directly as ammonia into the surrounding water, right? And as we learned from Dory, you can just swim away. And what I found particularly fascinating is that for animals to conquer land, that other solutions had to be found or devised in order to be able to get rid of this nitrogen. And so in mammals, the solution has been the urea cycle, converting ammonia into urea, which then can be excreted in the urine, for instance. But birds and other animals have found other solutions in terms of making uric acid. So it was in the 20s and 30s that people were working on trying to figure out how this ammonia was converted into urea. And this figure comes from Hans Krebs, who was the first to describe what the urea cycle, but what he called, and we'll see shortly why it's appropriate, the ornithine cycle. Now, our understanding is very different from this one, but as you can see, even in this very early diagram, it shows, a carbon from a CO2 and 2 amino groups that go to make this molecule urea, as you can see on the left here. So the urea cycle has 2 roles. Its main role in mammals is to convert ammonia into urea. But the other is that it makes arginine, as you can see here in basically the second to last step of the urea cycle. And this is an amino acid, a protein amino acid, as you all know. So as we talked about, ammonia is a waste form of nitrogen, which comes from protein degradation. And the urea cycle itself requires this coordinated function of 6 enzymes and 2 transporters. We'll get into them individually in a minute. So a defect in any one of these transporters or enzymes may cause a block in urea cycle function, and that can result both in a buildup of ammonia, which is a potentially common feature of all of the urea cycle disorders, but also arginine then becomes conditionally essential, right? So you will have a decrease or absent arginine production, and then you would, as a result, need to get it from the diet. So this is um, a schema of our understanding of the urea cycle disorder right now, but it's somewhat lacking in some ways, but appropriately represents all the components that we're going to review or discuss today. But sort of with that in mind, I do want to go. This is a diagram I particularly like from Lippincott that goes through in a little bit more detail these various steps, and I just want to share with you sort of this is how I think about the urea cycle, and wanted to share this with you. So it starts in the bottom right-hand corner here, and if you can see, there's CO2 plus ammonia plus 2 ATP, right? In the plus 2 ATP. So in the very first reaction of the urea cycle, one cycle of the urea cycle takes 3 ATP, and 2 of them are needed here. So it is certainly the most energy-intensive step of the urea cycle, and may very well explain why in disorders of energy deficiency or otherwise that you can get hyperammonemia, because this step may be most compromised. So in this first step, right, that you're getting this carbon and nitrogen and then phosphate group from the ATP to make carbamyl phosphate. And as you recall from the diagram of urea that I showed earlier, we're almost done, right? All we need to do is replace this phosphate group with another amino group, and that's urea there. So basically all the other steps of the urea cycle are there to accomplish that task. And so that obviously the next step in the urea cycle here is the OTC enzyme that takes the carbamyl phosphate, adds it to ornithine to make citrulline. As I shared earlier, Hans Krebs called this the ornithine cycle. And I think that's a more apt name, really, because it is ornithine that is doing the cycling. So to illustrate that, what I'm going to do is I'm just going to put a box around ornithine, because it's what you'll see is that we're going to use it as a scaffold to make urea. And at the very end, it comes out untouched, right? It gets reconstituted at the end, right? So, okay, so every time there's ornithine, I'm just going to put this blue box around it. You can see what we're trying to build at the top here. Citrulline needs to get transported out of the mitochondrion. Then what we're doing is we're finding the other nitrogen that to make urea that comes from another amino acid, aspartate. Once again, the carbon skeleton I'm just going to cover up, because as you'll see shortly, it's going to leave, right? So we're going to combine citrulline and aspartate with this step, arginosuccinate synthase. This is the one other ATP that gets utilized here to synthesize arginosuccinate, and then we cleave off this not needed carbon skeleton, right, in the form of fumarate, which then makes arginine. As you can see at the very top there, we're almost done. We're going to cleave off this compound, which is now going to be urea, with enzyme arginase, and it makes ornithine again, which then gets transported back into the mitochondria. Other thing I want to highlight is that all of these enzymatic steps are present in the liver. And really, so this is when we say this is the urea cycle, really it's the hepatic urea cycle. But some of these enzymes are expressed in other parts of the body. So I just want to show you here again each of these enzymes, their chromosomal location, and as well as the tissue expression. But I want to highlight that some, but not all of the enzymes are expressed in other tissues in the body. So for example, here, these first 3 are in the intestine, the next in the kidney. So what that looks like is that if you want to think of it that way, that there's an intestinal urea cycle where the goal strictly is to make citrulline, which then gets exported from these intestinal epithelial cells into the portal circulation. Citrulline is not modified by the liver, so it goes through the first pass and then it goes into circulation, where it then goes to the kidneys, which are primarily responsible for the biosynthesis of arginine in humans. So the kidneys have these 2 other enzymes, ASS1 and ASL, where it makes arginine. Urea cycle disorders as a whole, so how common are they? About 1 in 35,000 here. We have numbers from a variety of different sources, but I want to highlight the most common of them, representing at least half, if not more, of UCDs is OTC deficiency here. So I want to talk a little bit about how these disorders present. As I shared earlier, the common feature of these conditions is the elevation in ammonia that can be present. Ammonia is bad. It causes brain edema. Essentially, as soon as ammonia gets into the CNS, it is converted into glutamine. As you can see, that has a wide range of effects, not only causing cerebral swelling, but also impairing neurotransmission. And so has, again, a variety of different clinical symptoms that can occur as a result of acute hyperammonemia. This diagram just shows some of the differences in neonates that obviously have a much more narrow kind of way of expressing the hyperammonemia. You will typically see early symptoms that include poor feeding and vomiting, lethargy and irritability, respiratory distress, hyperventilation and respiratory alkalosis. And of course, acute encephalopathy as the cerebral edema gets worse. I also want to highlight that we've learned that neonates, well, and adults as well, can have subclinical seizures as a result of hyperammonemia, which is to say that these are not necessarily convulsive in nature. And so, with that in mind, it may be important in any children that have or adults that have hyperammonemia that they are monitored with EEG because they may be having seizures that are not recognized. Infants, children, and adults, again, have other, better ways or other ways of expressing the response to hyperammonemia, and this may be in the form of behavioral changes, psychiatric manifestation, again, quite common. They may also have their first presenting signs may be the liver dysfunction as well. Like with other metabolic disorders, there's a general rule: residual enzyme activity predicts, the age of onset and the severity of clinical symptoms, right? So obviously, if you have no enzyme activity at all, then immediately as you're ex utero, you're going to start building up ammonia and potentially developing symptoms. If you have normal enzyme activity, you should have no symptoms at all. And then somewhere in this middle here, and it's quite a broad range, that if you have partial urea cycle enzyme activity, then that allows you to escape the newborn period without hyperammonemia. But then as a result of some confluence of events that may occur later, may result in presentation. So again, in neonates, you have near absent enzyme activity. But not all UCDs are identified by newborn metabolic screen. I think there's a misconception that these are all picked up, but as many of you may know, that there are really only two of the urea cycle, canonical urea cycle disorders that are on the core RUSP, arginocytosinic aciduria and citrullinemia. So you're obviously missing the most common of these disorders, which is OTC, for instance, but some of the others are also, not present and cannot be picked up by newborn screening currently. We already talked about sort of this late onset or childhood or adult presentation, the asymptomatic group. I want to highlight that certainly more recently has been more work on what I'm calling posse-symptomatic OTC deficiency, which is to say that there are patients who have some but not all of the features of OTC deficiency that may not necessarily have frank hyperammonemia or evidence of impaired nitrogen metabolism based on biochemical tests, but do have clinical symptoms that may include abnormal executive functioning or memory, for instance. So how do you diagnose urea cycle disorders? Again, in brief, I'm going to kind of tier these in terms of laboratory tests that you'll get somewhat immediately versus a little bit later, right? So obviously you'll be getting an ammonia level that might be elevated, obviously a clue that your patient has a urea cycle disorder. as I talked a bit earlier, you can get a respiratory alkalosis as a result of the tachypnea in this condition. Oftentimes you'll see a disproportionately low BUN. And so by that, I mean that, it doesn't necessarily have to be below the normal end of the range, but, potentially in a dehydrated patient, you'll have a lower normal BUN, which is, not something you would typically see in another patient who does not have a UCD. The other thing too is that you may also observe abnormal liver enzymes. Typically, the coagulation profile tends to be slightly abnormal, not in any range typically that would require intervention, but you will see this typically first, followed by alterations in the transaminases. So, depending on your center, these top labs will come first, and then you will get some of these other lab results, sort of within a day to a week later, that would be important for the diagnosis of urea cycle disorders. Those include the plasma amino acid profile, the urine organic acid profile, plus minus orotic acid, which you could see as either part of the urine organic acid profile itself or ordered separately. Of course, you can always get DNA-based testing and enzymatic testing as well, although this to some degree has fallen out of favor for most disorders. It requires a liver biopsy, and this is done less and less frequently. And in fact, in North America, I don't know anywhere currently right now where they're offering UCD enzyme analysis of liver samples. That plasma amino acid profile is tremendously important, and one of the things you'll be looking at is citrulline, which is situated right here in the middle of the urea cycle. And so it can help differentiate between the disorders. So if your citrulline is low, it would suggest that your block is upstream. So these proximal disorders, urea cycle disorders, if you will, whereas if they're elevated, it might suggest that you have a downstream block that is causing an accumulation of citrulline, or the so-called distal UCDs. I just wanted, I'm not going to go through in detail here, but just to share with you, that there's somewhat of an algorithm to go through depending on what you're seeing on your amino acid profile that would help you identify which urea cycle disorder you should be thinking about. Of course, you don't always know with a patient coming in that they have a urea cycle disorder. So kind of in a broader schema, that given that there are many different conditions that can cause hyperammonemia, again, some of the things you can identify either on amino acid profile or organic acid profile that would point you or help you develop a differential diagnosis. In the urea cycle disorders, for the most part, an elevated glutamine is also observed in tandem with hyperammonemia, and that the reason for this is this following axis, as you can see here, that alpha ketoglutarate can combine with ammonia to make glutamate, which again combines with another ammonia to make glutamine. And so just stoichiometrically, as you have more ammonia in your system, it can effectively result in the production of more glutamine, again, measurable in that plasma amino acid profile. So I want to go through some of the, relatively quickly here, some of the individual urea cycle enzymes, as well as the disorders that are caused by them. And we'll start here with CPS1. So CPS1 is the first and rate limiting step of the urea cycle, and it in fact is apparently the most abundant mitochondrial protein. It has an essential allosteric activator, which is an acetylglutamic acid or NAG, and that well, teleologically anyway, the thought is that this provides a mechanism to rapidly change flux through the urea cycle, right? So again, it is the first and rate limiting step of the urea cycle. I think this is true in a lot of biochemical pathways, that the first step is the one that controls flux through that pathway. So PDH, for instance, is the rate limiting step of the Krebs cycle. And so this is much similar. And so, in a circumstance that you may need to rapidly change flux through the urea cycle, this potentially could be achieved by changing the amount of available NAG, right? So this does not require differences in, transcription or translation, for instance, and can be achieved pretty quickly. So because this is the first, biochemical step, there are no nitrogen intermediates that are produced. So as a result, it's one of the most severe UCD. Ammonia levels tend to be markedly elevated in those that have a null CPS1 activity, and a result, of course, in elevated ammonia and glutamine, as I previously mentioned, as well as low citrulline. There are some patients with CPS1 deficiency that have single amino acid substitutions that might be responsive to cofactor supplementation. And again, as you know, or you've already discussed, that this is a common paradigm in biochemical genetics, that pharmacological supplementation of a cofactor may help with stability, et cetera, and you may get improved enzymatic function from this. I saw, is there a question or comment here?
Yeah, it's actually a good question, but I
Yeah,
was gonna
yeah,
wait
sure,
until
sure.
there was a pause in the action. So, hold on, let me get my glasses on. Uh, Dr. Alsamak asked, he's got a 20-year-old patient with hepatocellular carcinoma and Tylenol intoxication. She had hyperammonemia that dropped with dialysis and scavengers. Basically, a clinical picture that looked a lot like urea cycle disorder, but with hepatocellular carcinoma. It included erotic aciduria, uracil in the urine, low citrulline, high glutamine. He's asking if there's any way to tell if it's a primary urea cycle disorder or a phenomenon seen with hepatocellular carcinoma. He didn't say fibrolamellar, but that's the type that I typically think of when I think of this phenomenon, hyperammonemia with hepatocellular carcinoma. But do you have any advice for how to distinguish the two, or know whether there's maybe both are present?
Huh. Okay. Okay. Apologies. Was there? Uh, can you share whether there's? I mean, obviously, molecular testing. Did I miss that, or?
He didn't say. So I would definitely go for
Uh,
molecular testing, but if it's negative in 15 to 20% of cases, then
no, I mean, I certainly would. So I don't know, based on this, that it's, um, that I mean, I think I would start with that. Uh, you know, but I appreciate that this is certainly a challenging scenario. To be able to make that differentiation. I also appreciate what we haven't gotten there yet, but to Dr. Baker's point, that there's a certain proportion of patients that have OTC deficiency that have no, that clinically diagnosed OTC deficiency, but that who do not have um, molecular findings consistent with that, and that these might be part of within intronic regions or regulatory regions, and that's something we're currently working on. Maybe if you don't mind, Dr. Al-Samak, perhaps we could chat at, following this, perhaps in a little bit more detail, or even after this lecture, in a little bit more detail about how best to address this. Thanks. But again, a really challenging, clinical scenario here. Certainly, the molecular testing is, I think, something would be potentially helpful. Great. Okay. So, yeah. So, okay, perfect. So next, I want to talk about NAG synthase. We talked about CPS1 and how N-acetylglutamate, as you can see here, is the essential allosteric activator of CPS1. That this N-acetylglutamate is produced by this enzyme NAG synthase, and so such that NAG synthase deficiency results in an absence of N-acetylglutamate, and it results in a functional CPS1 deficiency. So the biochemical phenotype is going to be near identical to CPS1 deficiency. I don't know why I said near identical. It's going to be identical. to CPS1 deficiency, and that NAG synthase is also is secondarily inhibited by organic, in the organic acidemias, for instance, propionic acidemia, and potentially the reason why you observe hyperammonemia in the organic acidemias. This disorder also is treatable with carglumic acid. And your question may be, why are we giving carglumic acid instead of N-acetylglutamate or N-acetylglutamic acid? The reason is this compound, if you give it orally or IV, is metabolized by acyl aminoacylases, and so it doesn't actually reach the liver where you want it to. And fortunately, there's this alternative compound, N-carbonyl glutamate, that is almost similar in structure as shown here, that is not metabolized and that can reach the liver. There are a number of different compounds, as it turns out, that can serve in the same function, but carbyl glutamate, of the ones we've seen so far, appears to be the best, or have the greatest affinity for the enzyme. OTC is the next enzyme in this pathway, and is an X-linked gene as a result of deficiencies in X-linked disorder, so that males who have a variant are more commonly symptomatic. Females with will only typically become symptomatic with skewed lionization, but once again, there's this cohort of posse-symptomatic females that appear to have executive functioning deficits without evidence of elevated ammonia levels. The biochemical features would include elevated ammonia and glutamine and low citrulline, but of course, elevated orotic acid, as we kind of discussed in that case earlier. And you may be asking, why is orotic acid elevated? Well, so if you consider the block the block here is in the OTC enzyme, it is upstream of carbamyl phosphate, which is made by this enzyme CPS1. So if it is called CPS1, that must mean there is a CPS2, and so here it is in the de novo pyrimidine biosynthetic pathway. And so somehow, and again, this is not fully understood, that this carbamyl phosphate makes its way out of the mitochondria into the cytosol, where it can then be converted into orotate. This can become even more obvious by performing what's called an allopurinol challenge, and blocking these downstream enzymes such that the elevations in orotate are more obvious, and can be measured in a 24-hour urine sample. For those of you who are following this lectureship series with the purpose of exam preparation, once again, this is an X-linked condition, and it is only one of the urocycle disorders that is such. And as a result, it is the only urocycle disorder in which you will commonly see de novo variants that cause disease. That this proportion is approximately 20% de novo versus 80% inherited. And there's a variety of different reasons for this. One of them being sort of differential mutation rates in sperm and egg, for instance. But I would say, again, for example purposes here, I would stick with this idea that in Mendelian conditions with virtually no reproductive fitness, that 2/3 are inherited, 1/3 are de novo. I also want to highlight this recent work here that from our group, just perhaps better characterizing this posse-symptomatic group, that those apparently who are asymptomatic carriers of OTC, that not all of them are always asymptomatic. And this article goes through a discussion of, again, some of the executive findings, some of the neuroimaging differences, as well as how many of those actually went on then to have developed hyperammonemia. This next enzyme in this pathway is the ASS1 enzyme that causes citrullinemia type 1, which, once again, elevated ammonia and glutamine. You'll see this almost in every slide here, but also markedly elevated citrulline levels. The word citrulline comes from citrulus, which is Latin for watermelon. because citrulline was first isolated from watermelon, and watermelon has very high concentrations of citrulline in all of its parts. Other species in the same family, like cucumber, for instance, do have a slightly elevated citrulline, but certainly not anywhere near the concentrations of watermelon. And watermelon consumption can result in temporary elevations in plasma citrulline and arginine. And there's at least one publication of, that goes through a child with developmental delays who had an amino acid profile where they didn't have citrullinemia, but ate a lot of watermelon, and so their citrulline levels were elevated. We don't think that citrulline is toxic in any fashion, but at least in this publication, there's some discussion of correlation between, controlling for everything else, correlation between elevations in citrulline and neurocognitive outcomes. So maybe there's more of a story there. Also want to highlight this article that, again, I think has some methodological flaws, but would suggest that there are some ASS1 carriers that have slightly elevated blood citrulline levels. And I think this is becoming an issue more and more now because of newborn screening, where you might identify some kids based on newborn screening that have, slightly elevated citrulline levels, and subsequently when, if or when you do molecular testing, you only identify one variant. But I do want to, make you aware that there is such a publication, although I think potentially some of these patients may have, sort of a non-coding or non-identified variant on the transallele. This next enzyme is ASL, or arginosuccinic lyase. And this is particularly interesting, or the deficiency of disorder is particularly interesting because there are some other clinical features of this condition that include trichorexis nodosa, in part because there's a lot of arginine in hair, but also because of hypertension as a result of impaired nitric oxide metabolism, which I'll get to in the subsequent slide. Once again, high ammonia, elevated glutamine. You will also see elevations in arginosuccinate and its anhydrides on your plasma amino acid profile, and this may also then back up to result in mildly elevated citrulline, which is the marker used at least by many states on newborn screening. ASA, arginosuccinate, is an amino acid and can be identified, obviously, with an anhydrin, but importantly, it is not reabsorbed by the kidneys. There's no way of absorbing the ASA back, which means that almost all of your ASA is going to be excreted in your urine, and as a result, the urine concentration of ASA is going to be more abnormal than in the plasma. So if there's ever a patient where you're getting an amino acid profile that you're not entirely certain whether you're seeing an ASA profile, oftentimes a urine plasma amino acid profile, which is, again, really helpful, but it's specifically in this condition may be utilized in order to be able to identify elevations in ASA. This diagram here just to show you that the link between the uricycle and nitric oxide synthesis. So this will occur, for instance, in endothelial cells where there is that you require ASS, ASL, as well as nitric oxide synthase that's going to convert arginine to citrulline and release nitric oxide. Right. So if the step just prior, which is ASL here, is dysfunctional, that might impair nitric oxide synthesis and subsequently result in hypertension. Great. Okay. And then the point here about the ASA and its anhydrides, for some of the lab folks here, that so you're aware that ASA colutes with leucine, and its anhydrides colute with beta-alanine and GABA. So this particular pattern of elevated leucine with beta-alanine and GABA, which you should really never see, in a plasma amino acid profile, would be suggestive of this condition. This next enzyme here is arginase 1, which is a little bit unique because it is as distal as it is. Hyperammonemia is an uncommon but certainly possible feature of the condition. That you'll see markedly elevated arginine, as well as some other features that include peripheral neuropathy, intellectual disability, and seizures. And in this phenotype in part is as a result of the fact that elevated arginine increases production of guanidinoacetate acetate. So you might have some features in common between arginase deficiency, for instance, and GAMP deficiency. We've covered all the enzymes. I want to talk briefly about the two transporters. The first is citrulline, the citrulline transporter, which is its gene is SLC25A13. It codes for this aspartate glutamate countertransporter, and you will see elevated citrulline. There are some different phenotypes of this condition that include intrahepatic cholestasis in neonates, failure to thrive in dyslipidemia in those of juvenile age, and then in adolescence or in adulthood can result in recurrent hyperammonemia or the so-called citrullinemia type 2. Variants, disease-causing variants are most common in the Asian populations, with the carrier rate there in East Asia or Southeast Asia of 1 in 50 to 100. So, I think there's an argument to be made here that this potentially globally the most common UCD and not OTC. Particularly interesting here is that there's a couple of recurrent variants in the Asian population, and so I think there have been some theories that there may be some selection bias, although the exact selection bias is unclear at the moment. The other thing that's particularly interesting is that whereas for some of the other disorders, there's a pretty clear correlation between residual enzyme activity and phenotype, and I think that tends to be less clear here for citrin deficiency. Another unique feature is that unlike the UCDs, that patients with citrin deficiency should be treated with a high protein, high fat, and low carbohydrate diet. And this appears to be the case because there's impaired redox, so NADH, NAD redox in this condition that tends to be exacerbated with a higher carbohydrate diet that's typical of UCDs, and that patients actually self-select a high protein, high fat diet. The other transport is the ornithine transporter, again coded by this gene SLC25A15. So, given where it's situated here, will result in elevated ornithine levels, typically in the 300 to 500 micromolar range. But unlike with ornithine aminotransferase deficiency, what typically has much higher ornithine levels, this condition, HHH deficiency, does not cause the type of ocular abnormality that you'll see with OAT deficiency. So there, in some early childhood onset forms, you'll get gait abnormalities and spasticity. There is a common founder mutation in the French Canadian population, as shown here, and there's also chronic liver involvement that's I think better discussed in this publication. Okay. So again, very brief overview of the different enzymes, some of the features of the condition. I want to talk briefly about the management of urea cycle enzymes. And as a general rule, sort of these are the principles that I think about in terms of the management here. So let's start at the top, which is to reduce or stop the influx of excess nitrogen, or stop production of ammonia. So, this nitrogen comes, in the blood will really only come from two sources. It'll be exogenous, in other words, dietary. So you're providing this to patients. But alternatively, it could be endogenous. You have to keep in mind the body has a lot of proteins, in particular in muscle, and that during periods of catabolism, these could be released and utilized. Their carbon skeleton utilized for ATP production, and as a result, release this excess amino groups in the form of ammonia. So that process has to be reversed or reduced in some fashion. And typically the way to do that is to provide excess non-protein calories, or dextrose, or intralipids, et cetera, that may help reverse that process. And in the scenarios where patients might be hyperglycemic from getting too much glucose, that you may even start an insulin drip that has some anabolic potential as well and promote anabolism. So simultaneous to that, you also want to consider other ways of removing nitrogen outside the urea cycle. Easiest way to do that, at least conceptually, is to just take blood out of the body, take out the ammonia and put it back in, right? So continuous or intermittent forms of hemodialysis or hemofiltration. And certainly that's an option that's always available to you in a circumstance where ammonia levels are markedly elevated. But obviously keep in mind the risks associated with this and the time it takes to connect the patient up to this machinery. There are also, as you may know them or colloquially know them, ammonia scavenger medications that don't really remove ammonia per se, but that they remove non-essential amino acids. In other words, amino acids that the body can reconstitute from carbon and ammonia, or carbon skeletons and ammonia. And these medications work stoichiometrically, which is to say that every mole of these medications that you're providing then will eliminate a certain number of moles of nitrogen. So the two that we have currently available to us are benzoate and phenylacetate. So benzoate combines with glycine to make hippurate or hippuric acid, whereas and so glycine has one nitrogen as shown here. Phenylacetate, on the other end, combines with glutamine to make phenylacetylglutamine. Again, so you're getting rid of two nitrogen. I was always taught that the enzymes that perform these two conjugations are different. The more I've looked into this, I'm not sure about this, but certainly, that these medications have been combined and work effectively together. Phenylacetate itself is really noxious. This precedes my time in terms of when this was used. Just pure phenylacetate, orally or enterally as a medication. So somebody had the bright idea of saying, okay, well, why don't we add two carbons to make phenylbutyrate, have it go through one beta oxidation step. And as many of you know, there is oral phenylbutyrate that is available, and I think a little bit more palatable. But then even beyond that, someone had this idea of turning this into an oil, right? So having having a glycerol backbone and three phenylbutyrate molecules that makes this oil glycerol phenylbutyrate that is, I think, certainly easier to administer from a volume perspective than just sodium phenylbutyrate. So this basically is a pre-prodrug, if you want to think of it that way, right? So going on the right-hand side here, the glycerol phenylbutyrate has to first undergo, ester hydrolysis to make phenylbutyric acid, which undergoes one beta oxidation step, which I think is done by MCAD to make phenylacetate, and then is conjugated with glutamine to make phenylacetylglutamine, and these are excreted in the urine. Both compounds, the phenylacetylglutamine and the hippuric acid, are excreted in the urine very efficiently, but again, keeping in mind your patient circumstance, certainly if they have renal dysfunction or failure and they have oliguria, then you should consider take that into account. As shared, these medications may be administered together in the form, as many of you know, and at least in the United States, in the form, IV form called Aminol, but you can certainly give them orally or enterally together as well. There's no direct head-to-head comparisons of these two compounds in UCD patients, but I will point you to this study of phenylbutyrate and benzoate that was performed in healthy adult volunteers. In terms of management of these conditions, I always like to point out that you really need to do these two steps simultaneously. And this is really important. One analogy that I like to utilize is that one of an overflowing sink to represent what's happening with an acutely hyperammonemic patient. Right, so you have this excess influx of water or ammonia, if you will, and that you want to reduce that in some fashion, but simultaneously you want to try to get rid of water from your sink basin, and that if you only do one of these steps, which is you're frantically bucketing out water without turning off the tap, then you're not making any progress. And similarly, if you are just turning off the tap, then this water level will continue to remain high and will take longer to descend. I want to discuss arginine and citrulline supplementation in these disorders as well. So the example I like to best use here is what happens with ASL deficiency and why arginine supplementation is important in this condition. We already talked about how arginine becomes conditionally essential in disorders where you have a block in the urea cycle. But conceptually, if you think about it, essentially what's happening is when you have a block in the urea cycle, you are converting this cycle into a linear pathway that goes from arginine, in this case, all the way to arginine. So I think when you lay it out in this fashion, it becomes much more obvious what happens if you don't have any arginine, right? So you're going to eventually become arginine deplete, as all of this arginine is going to make arginine. But the other thing too is that once you replace arginine, then it also allows every one of these other steps to then occur, right? So it allows the ammonia to get incorporated and allows this nitrogen from aspartate to get incorporated to arginosuccinate. As we discussed, arginosuccinate is very effectively excreted in the urine. It has 4 nitrogens, so this is a great way to be able to get rid of ammonia. I'm making this trade of ammonia for arginosuccinate, 1 mole for 1 mole every day, all day, every day in patients with acute hyperammonemia. That having been said, there's at least more recently evidence to suggest that we should, at least in the chronic setting, get away from high dose arginine. This is in this publication, this was a randomized crossover trial evaluating high dose, in other words, 500 milligrams per kilogram per day of arginine versus low dose arginine with scavengers. Those who are on low dose arginine with scavengers had improved hepatic function tests in this study, suggesting that maybe there is a more toxic role, for instance, for arginosuccinic acid or some other downstream compound that again would suggest that chronic therapy should be more oriented towards a low dose arginine plus scavengers that would divert nitrogen away from the urea cycle. In OTC, the same idea here, but instead of giving arginine, which is further down, you'll give citrulline, which then at least allows 1 nitrogen to be incorporated from aspartate before you keep going along. Ornithine typically is not that elevated in OTC deficiency as it gets broken down by OAT. So we discussed these first 3. I want to briefly discuss improving enzymatic function. We talked about how carglumic acid might be useful, obviously in NAG synthase deficiency, but potentially in some forms of CPS1. From a chronic perspective, liver transplant might be a way to achieve normalized hepatic enzymatic function in patients with a urea cycle disorder. And more, there are clinical trials now for, different types of enzyme replacement or mRNA therapies or gene therapies that certainly in the works, and you're going to hear more about in the upcoming years. The last thing I want to touch on here is the treatment of intercurrent illnesses, which is really important. And in that process, also consider other triggers of hyperammonemia. Some of you may have heard of a couple of years ago, there was a star high school quarterback who passed away, and as it turns out, he had a urea cycle disorder. He had surgery. He received corticosteroids. And so there are some of these things are known triggers for hyperammonemia in UCD patients, and likely the explanation why he was able to make it so long without any clinical symptoms. And then this confluence events of fastings for surgery, corticosteroids, et cetera, triggered, an hyperammonemic event, unfortunately. And so these are common triggers of hyperammonemia, illness, poor diet, missed medications, stressful life events. And then within this there's some other triggers that include prolonged fasting and surgery, as we discussed. Gastric bypass may be a type of surgery that's particularly prone to, causing hyperammonemia. Also, unfortunately, the model of care for the peripartum and postpartum period often involves prolonged periods of fasting. So again, a common trigger of hyperammonemia in UCDs. The other thing that occurs postpartum is sort of the involution of the uterus that is, has to be quite large to sustain a growing infant, growing fetus, but then, immediately postpartum has to shrink back to its typical size, and that also releases a tremendous amount of protein. Menstrual cycles may be certainly a common trigger, or it can be a trigger for hyperammonemia in UCDs, as well as these medications that either promote catabolism or might impair the urocycle in some fashion. So I'm gonna perhaps stop there. I see some things in the chat here with respect to, oh, is there any questions? Okay, sorry.
No,
I saw some comments, I wasn't sure. Okay. If there are none, then I'm just gonna go straight into MSUD here, and maybe if there are other questions, we can discuss those at the end. This will be a little bit shorter part of the discussion here, but so what is maple syrup urine disease? Many of you already know that stands for, MSUD stands for maple syrup urine disease. It is caused by a partial or complete lack of the branched chain keto acid dehydrogenase, or BCKD complex. And this enzyme is needed in the breakdown of branched chain amino acids. Your next question is, well, what are those? And so these are the branched chain amino acids. These are essential amino acids, in other words, not made by the body, and they have a branched side chain, right? As shown here. They're somewhat similar, visually in appearance, but also potentially, to terms of this makes differentiation by, some methods like tandem MS might be somewhat difficult. So this is this loose, branched chain amino acid degradative pathway. And this is where the BCD complex is located within this pathway. And so a deficiency of this enzyme complex will cause elevations of the amino acids, but also their keto acids, which is part of the reason this condition called is maple syrup urine disease. The keto acid of isoleucine also can go on to make alloisoleucine, which is elevated in this disorder, and I think is pathognomonic for maple syrup urine disease. However, the toxic amino acid in MSUD is leucine, and the one we are most trying to control within the plasma and within the body. You may be wondering why it's called maple syrup urine disease. There is another byproduct of isoleucine called sotalone that gives it its characteristic sweet or burnt sugar odor that can be present in urine, as the name suggests, but also really present in earwax as well. So one of the methods of facilitating diagnosis is sniffing ear of an infant who's encephalopathic to get a better sense of whether they might have MSUD. This particular odor, though, is not unique to this disorder. It appears also to be found in some spices, including fenugreek. And so there have been several publications of this pseudo maple syrup urine disease as a result of fenugreek ingestion. The BCKD enzyme complex has 3 different subunits, or has different subunits, including E1 alpha, E1 beta, E2, and E3. And so obviously that deficiencies of any one of these can result in MSUD. This is strictly an autosomal recessive disorder in all of its forms, with an incidence of about 1 in 185,000. I don't know if I discussed it later, but just to point out that this E3 component is also shared with some other enzyme complexes, and as a result, that particular disorder is quite different than the typical patient who has MSUD. Most patients who have classical MSUD or typical MSUD will have variants in E1 alpha, beta, or E2 genes. Even with MSUD, again, depending on the mutation type, you will have potentially varying phenotypes. The classic or severe is the most common form and will, will result with a neonatal presentation. But there are those who have an acute intermittent forms, in other words, that they don't present neonatally, but will have some type of acute presentation. And within this group, amino and keto acids might actually be normal between episodes, so you're only going to diagnose them during that acute decompensation. And there's some that have this intermediate or subacute form that will have developmental delay, hypertonia, and failure to thrive, but not really any type of acute decompensation. Why is high blood leucine toxic? Well, similarly to ammonia, it causes brain edema. And as you can see here with the imaging of a patient on the right-hand side who was an MSUD patient in crisis. So much similar to patients that have hyperammonemia, this brain edema will result in a spectrum of features that includes poor feeding and vomiting, irritability, fatigue, potentially neuropsychiatric symptoms, lethargy that develops into coma, abnormal tone of movements, seizures or ataxia or other neurological presentations. MSUD, fortunately, is on the recommended uniform screening panel, so there are hopefully patients who are, we hope that most patients are identified that way, although obviously it is possible that patients may present acutely even before the results of your newborn screening are back. You will get diagnosed with MSUD after you confirm elevations in the branched chain amino acids, as well as I said, as alloisoleucine, which is pathognomonic in this condition. Patients who are presenting with acute decompensation may have evidence of hyponatremia. I'm not sure I know the origin of this, but this is a feature of patients who are with MSUD who are in crisis. Beyond the plasma amino acid profile, certainly you could go on to sequencing of BCKD genes in order to be able to further confirm your diagnosis of MSUD. Vitamin B1 or thiamine is, if you will, a cofactor to this to the BCKD complex, and so there are some patients, there are a limited number of patients in whom high dose thiamine therapy can actually improve BCKD function and reduce or eliminate the need for any special dietary modification. But once again, similar to with PKU, this certainly can be really challenging to interpret results of this type of challenge. In terms of management of acute crises of MSUD, these same principles, if you will, I think still apply in terms of what to do and this analogy of overflowing sink. The only thing is here is there's not really any alternative medications to give. So, really what we're trying to do is, these amino acids really only have two fates. Either they go on to make protein or they're degraded. And so the options here are basically how do we promote protein synthesis in patients in acute crisis? And this is done by providing all the amino acids except leucine. And so this may include a branched chain amino acid free formula by mouth or by tube. If you have access to parenteral nutrition, so branched chain amino acid free TPN or IV solution, that'd be great. And then you will also want to supplement with isoleucine and valine as these may become low in patients who are being treated and are making more protein. Like you basically do not want these two amino acids to be the rate limiter in terms of protein synthesis. But they may also, to some degree, impede leucine transport into the brain as well. So similarly to doing all of these things, you also want to reverse catabolism that will involve giving a lot of calories. And if not tolerating a type of enteral feeding, you may choose to give IV calories, typically dextrose at high concentrations, and keeping in mind to keep these solutions, at least normonatremic or hypernatremic, if you will. Again, for the reason I described before, which is that many of these patients can be hyponatremic, and so I would avoid solutions that are hypotonic in nature that may exacerbate the brain edema. I think there's been a limited success with mannitol and diuretics in this population. And then once again, if you have no other tools, you can hemodialyze your patients. This chronic dietary management for this condition would involve providing an exact amount of regular protein. By that, I mean natural or intact or complete protein. There's some teams that may choose to, and I think it's probably more exact way of measuring the exact amount of branched chain amino acids that patients are receiving daily. And as you know, there are synthetic formulas that will have everything except isoleucine, leucine, and valine in it that will provide all the calories, essential fats, vitamins and minerals. And then on top of that, you would supplement valine and isoleucine in order, to better control your levels. Given everything here, you may also add protein-free foods that would provide additional calories and nutrition. These are the target branched chain amino acid ranges as determined by GMDI, the Genetic Metabolic Dietitians International. I think by Delphi process. But in short, what you're trying to do is keep the leucine levels within the normal range, and as much as possible, try to keep this typical ratio, of leucine, isoleucine, valine of the physiologic typically would be 2 to 1 to 3 here. The thoughts behind keeping isoleucine a little bit higher, once again, may have to do with you not wanting this to become deficient at any point, but also potentially, competing with leucine in terms of transport into the brain. There are many patients with MSUD who have successfully received a liver transplant. The BCKD complex is a somewhat ubiquitous enzyme, so that liver transplant from an unrelated donor really restores only 9 to 13% of total body branched chain keto acid metabolism. So this is sufficient typically to allow a much less restrictive diet and prevent acute crisis, although there have been some that have occurred in patients post liver transplant. This is pretty rare though. And this ratio of branched chain amino acids tends to be relatively normal. So they tend to be twice the normal range, but in the typical normal ratios. I also lastly want to highlight that domino liver transplants have been successful in this condition, which is to say that, the patients with MSUD may receive a transplant from an unrelated donor, but that their livers then go on or then donated to another person who has liver failure of another type, not MSUD, and they're receiving the liver of the patient who has MSUD. The idea here is that once again that almost 90% of the branched chain keto acid metabolism remains intact in those individuals and is only deficient in their liver, but everything else in that liver is functional. So hopefully corrects the reason why they are getting the liver transplant. So great, perfect. So I think I've covered again this overview of these two disorders. It's difficult to squeeze everything into this talk, but want to thank you for your attention. And I also want to highlight that certainly for myself, there's no real acknowledgement slides per se, but that sort of what I'm sharing with you today is as a result of things that I myself have learned from others, other mentors, Mendel Tuckman and Marshall Summer and Mark Batchot and Andrew Grobman at Children's National, but also from the patient organizations like the NUCDF and the MSUD Family Association. And I would highly encourage you to be involved with those groups as well, as they're fantastic patient advocacy groups. So I'm happy to take questions.
Thank you so much, Dr. AhMew. That was great. Some awesome pearls in there too. The watermelon and citrulline connection always makes me smile. Questions for Dr.
It's really interesting because I think there's some that think it's really only within the seeds or only in the rind. This is not true. There are multiple studies that have shown citrulline is markedly elevated in any part of the watermelon. As a general rule, red watermelon tends to be slightly lower than the orange or yellow varieties, but citrulline is still really, really high in all of them.
That's great. We've had actually some of the elevations, slight elevations of citrulline on the newborn screen, maybe carriers, probably carriers, but I've been trying for years to find somebody who just ate watermelon before the screen was done and is breastfeeding, but haven't found one yet.
Well, so as I shared, there is one publication of, I believe it's a girl who had some delays, got amino acid as part of that workup, and had elevated citrulline levels, that came from watermelon ingestion. And then the subsequent part of that paper was, 4 guys cracked open a watermelon, ate it, and measured their amino acids afterwards. And I thought that's brilliant. That's the best way to get a peer-reviewed publication is to eat a watermelon. But yeah.
Yeah, there are a lot of calls and asks for the slide
Yeah,
set.
you bet.
So
No problem. I'm sorry that I don't have those to you yet, but I will make sure you get them.
yeah, no problem. If you send it to us, we can put it on the SIMD website along with the video presentation. They give us a little bit though, at least a few days. There was a question, any advice about increased detection of attenuated MSUD on newborn screens?
Yeah, I don't. Again, I realize there are some that would present that would only have biochemical abnormalities during periods of decompensation. So I'm not really sure how to pick those up otherwise. You know, uh, I know it's somewhat of a controversial topic, but certainly at some point when there is better molecular sequencing as part of newborn screen. I'm sorry that may be again controversial and blasphemous in some circles, but were we to get there, certainly that would be potentially a way of identifying those. I'm sorry, Dr. Elsimak, that I'm relying so heavily on molecular sequencing here, but I don't know that in it, sort of the intermittent MSUD forms that that there are any that there is a clear biochemical phenotype that could be detected by newborn screen. Oh, sorry.
I feel like we get questions from PCPs at least once a month about either an older kiddo that smells sweet or concern for developmental delay and intermittent MSUD. It's it would be nice if there were a better way to make sure we're most sensitively detecting it on newborn screen.
Yeah. So, what's the maximum dose of arginine and citrulline in type 1? Um, you know, I I think so. I think it would be in the range of chronically probably are in the range of 200 milligrams per kilo per day. I think acutely you may choose to give as high as 500 milligrams per kilo per day. Um, Once again, for the same reason, in the acute setting, it's possible that the arginine then goes on to make, allows some component of the urine cycle to function and make citrulline that is excreted not as well, obviously, because it is retaken up by the kidneys, but additional citrulline excretion may be beneficial in this disorder. Yeah, as a whole, though, I would probably titrate to arginine levels in those patients. Once again, with the thought that plasma arginine levels. Once again, with the thought that citrulline is likely not harmful, um, not toxic. So I don't have an active in vitro in vivo research program. I'm sure there's a list of animal models that we can get to. I don't have one though. Yeah, sure, you bet. Should I just put my email address in the chat? That's the best way to do that. I think I'm a little bit more familiar with the well, so for MSUD there are, I think, several mouse models. I'm aware that there's a cow model as well of MSUD that has been utilized by the group at the Hospital for Special Children in Lancaster. For UCDs, there's a variety of different mouse models, and I think at least one pig model of OTC deficiency. Yeah, I think if you wouldn't mind, I think what would be helpful here is perhaps if you wouldn't mind just email, because I think it's tough, to discuss very specific clinical cases here. Maybe if you wouldn't mind just emailing me those specific scenarios, and happy to kind of review or go through and give you my thoughts.
Some good questions and comments. More questions for Dr. AhMew. All right. I think we're going to wrap it up if there's nothing else. Thank you so much, Nick, for talking. It was a great lecture. Lots of thanks in the chat too. Thanks for everybody for showing up today. Have a great Labor Day weekend if you're in the US. Enjoy your day off on Monday. And yeah, we'll see you next week, Dr. Algarbawi. Thank you.
Again, thanks, Peter, and thanks everyone.
The website for posting slides is simd.org.
Gotcha. Okay, and I will send these to you right now.
Cool.
Thanks.
Yeah, thank you so much.
Take care.
Bye, guys. Bye, everybody.